actually start to exploit it in a way that will help patients with new therapies and drugs.
This is Dr. Les Beisaker, who's the current president of ASHG.
This as an activity that is completely separate from my day job, which is for the National Institutes of Health. So I'm here only for ASHG, and so please.
And what you think are the sort of the hot things happening in genetics right now, and I thought maybe this talk on the schizophrenia exomes might be a good one to ask about, because I wasn't sure. A couple things I wasn't sure about is they cited there have been lots of recent studies on genetics of schizophrenia, but this is, I guess, the first big exome study.
First very large exome.
Yeah, and I also wondered, is what they're seeing, have people done what they're doing for other diseases, or is this one of the first ones where it's
schizophrenia? And the results that we saw yesterday are very exciting, is that it's been a very challenging disease on which to work for a variety of reasons. My own personal work has been on a relatively simpler property or trait, and that's cholesterol levels, which is actually fairly straightforward. You can measure cholesterol levels, it's reproducible, it's quantitative, and so we were actually symptoms. How do genes influence the risk for schizophrenia? And it gives us clues as to how one might go about developing new treatments for schizophrenia.
What we're going to see here is a major paradigm shift in science. What we have been doing for over 100 years is going out and finding people who have what we think we understand is a disease. We gather those people up, we try our darndest to phenotype them as precisely as we can, and then we study the molecular. Schizophrenia and autism are the spectrum of one disease process. Nobody was saying that. The genomics, because we do high throughput genomics now, and we gather people, we can then align.
Trying to classify tumor based on those visual images, and in retrospect, we realized it wasn't actually doing a very good job.
Across these diseases, the treatment modalities will also be shared, which is, that's an incredible advance. Same thing.
Yeah.
Right?
No, that's
I
very
think
true.
you could answer that better than me.
So I study cardiovascular disease, heart disease, and a very strong risk factor for heart disease is blood lipids, so cholesterol, triglycerides, and so forth. And we had a fairly simple-minded view of things, I would say not even 10 years ago. There was the view that there's LDL cholesterol, the bad cholesterol, and there was a good cholesterol called HDL cholesterol, and then there was triglycerides, which was the fat content in the blood that we didn't really pay much attention to. We mostly focused on the good and the bad cholesterol. And what we have now found through genetics is that the bad cholesterol truly is bad, and whatever you do to reduce that bad cholesterol will be helpful and will reduce your risk of disease. What we have also learned is that the so-called good cholesterol, it's good in the sense that if you have high good cholesterol, it is protective, or at least it's associated with less risk, but it's not actually what's causing less risk. And so if you make drugs that increase the good cholesterol, they don't actually do anything. They don't work. And it just speaks to the complexity of what's going on underneath, and the simple associations we get by studying groups of people and asking, what's happening with people with high cholesterol, or good cholesterol, or bad cholesterol. It turns out that's the
efficacy, duration of efficacy for these gene therapies. Does it very much depend on the type of disease you're trying to?
A monoclonal antibody that you inject into the body, and then the antibody.
When is that?
Thursday afternoon.
Thursday afternoon
at 2 p.m.
2 p.m.
Fairbanks.
Which is a whole other level of therapy, right?
Yes.
And
now, but I've just finished writing a book that will be coming out later this month. That
gene editing is going to be added to that probably, because that's one of the tools in the toolkit. There's gene therapy, there's enzyme replacement therapy, there will be gene editing, there will be small molecule therapy. And it is a huge challenge to figure out which of those multiple modalities is going to work best for each of those disorders. And so you're going to see plenty of work by various groups of people who are at different stages, depending on the nature of those diseases and the rarity or commonality of those diseases, to figure out what's the best way to tackle each one of them, because they're often very severe disorders that manifest early in life with devastating consequences. But that's also an opportunity because newborn screening is such a well-established paradigm, in the States at least, that we have the opportunity to find these people, and we're currently primarily finding them by their metabolic phenotype. And an overly open, interesting question is, should we be either, instead of doing that, or maybe in addition adding to that, finding them genomically?
We had a session that looked at the versioning technology to actually be able to do genetic testing at the fetal stage in utero. So this involves what's known as cell-free DNA assays. You take DNA, or you isolate it from the mother's blood, but some of the fetal DNA leaks into the mother's blood. And so what you can do is you can take mom's blood sample, you can sequence all the DNA you find in there, and then you can subtract out mom's genome, and then whatever's left by inference is what came from the baby, from the growing baby. And you can look at genes that you are concerned about might be involved in exactly what you mentioned, inborn errors in metabolism. And there is the possibility now to actually diagnose these things even before the kid is born, even before a lot of damage is done, and know from the get-go that the issue is there, rather than what often happens now, which is the kid is born, and then in the few days or weeks after they're born, bad things start to appear, you don't know what's going on, and you have to do some sleuthing and figure out, and by that point, a lot of damage is already done.
Aspect of it, and finding a sort of how to regulate that and proper process. Do you ever see that as something that's going to become a uniform thing throughout the world? Because you see recent cases of the human-monkey chimera, where the doctor travelled from America to China to conduct that research. Do you think it's an achievable aim to get that as a fully uniform sort of regulation throughout the world, or is it something that might always have blocks?
Or everybody, I should say, to agree on one thing. I mean, that's just the reality of the situation. And, there's so many countries